A thousand-year-old shipwreck legend said exactly 7 couples survived. Every historian dismissed it. Every scholar called it a myth. Then scientists cracked open the DNA of an entire community, and the number that came back from the genome was 4 to 8 founding men. A children's bedtime story, repeated for 2,000 years, turned out to be approximately true. But that was not even the shocking part. The real shock was what happened when researchers compared this community's genetic blueprint to Jewish populations on the other side of the planet, and found the exact same formula hiding inside all of them. The same pattern, the same structure, exposed for the first time. By the end of this video, you will understand a genetic secret that connects a tiny fishing village on India's monsoon-battered coast to synagogues in Warsaw, Casablanca, Addis Ababa, and Sao Paulo. A secret that no rabbi, no historian, and no scripture ever revealed. Only a genome could. Make sure you watch this one all the way through, because the final piece of evidence ties everything together in a way nobody saw coming. There is a community in India that has prayed in Hebrew for over 1,000 years, kept the Sabbath longer than most European Jews can trace their own family names, and until recently, almost nobody could explain how they got there. Picture the inside of a synagogue in Alibag, a coastal town south of Mumbai. A Star of David is carved into the stone above the doorway. Women in saris are singing prayers in Hebrew, a language born 3,000 miles to the west, in a desert most of them have never seen. On the wall behind the congregation hangs a menorah. On the shelf beside it sits a framed photograph of someone's grandmother. The face in the photo looks entirely South Asian. The object next to it is entirely Jewish. 2 worlds in one room. India's Bene Israel community once numbered over 20,000 people at its peak, roughly the population of a small college town, yet large enough to make them one of the oldest continuously practicing Jewish groups on the planet. Despite that, mainstream genetics barely looked at them for decades. They were too far from Europe, too far from the usual story. Then DNA testing finally reached them, and what the chromosomes said about where this community actually came from did not match what anyone expected. Not the historians, not the rabbis, not even the Bene Israel themselves. The question is simple enough to say out loud, and explosive enough to have ruined careers. Are India's oldest Jewish communities, the Bene Israel of the Konkan coast and the Cochin Jews of Kerala, genuinely descended from ancient Israelite ancestors? Or are they Indian-born converts who adopted a foreign faith and nothing more? This was never just an academic debate. Israel's own rabbinical authorities spent years arguing over it. At one point, Bene Israel families trying to immigrate to Israel were challenged on the grounds that they might not be real Jews. Not culturally, genetically. Identity, citizenship, the right to marry under religious law, whether your children are considered part of the community at all. Everything hung on a question no rabbi could answer from scripture. It would take a genome to settle it. And when that genome was finally read, one strange genetic signature appeared inside it, a signature that linked the Bene Israel not only to the ancient Middle East, but to a pattern found in Jewish communities on almost every continent. That pattern will not make full sense until the very end of this story. Before the DNA arrived, there were plenty of theories. None of them held up. The oldest and most romantic explanation said the Indian Jews were descendants of the 10 lost tribes, the northern Israelite kingdoms deported by Assyria in 722 BCE, and never heard from again. British colonial writers loved this idea. It turned a small fishing community on the Konkan coast into a biblical mystery, a living relic of a 3,000-year-old exile. The problem is that no genetic study of any Jewish community, anywhere in the world, has ever confirmed a clean lost tribe origin. The concept is theologically appealing but scientifically unfalsifiable. You cannot prove descent from a group that left no verified genetic baseline. And the Bene Israel themselves never claimed a specific tribal identity. Their own tradition says nothing about Assyria. A second theory was more practical and, on the surface, more plausible. Trade routes between the Red Sea and India's western coast existed for millennia. Merchants carried goods, and sometimes they carried ideas. Maybe a group of Indian families simply encountered Jewish traders, adopted their religious practices, and became culturally Jewish without any biological connection to the Middle East at all. Pure conversion, zero migration. It sounds reasonable, but it makes a testable prediction. If the Bene Israel were purely converts with no Middle Eastern ancestry, their Y-chromosome profiles, the genetic markers passed from father to son, should look indistinguishable from surrounding Marathi-speaking Hindu populations. They do not. There is also a curious occupational detail. For centuries, the Bene Israel worked almost exclusively as oil pressers, a hereditary trade so specific it became part of their identity. Oil pressing mirrors ancient Levantine olive culture practices. It is an oddly narrow profession to borrow from a passing ship. A third camp argued for a much later arrival. Perhaps the community formed only 400 to 800 years ago, seeded by medieval Arab, Jewish, or Yemeni traders who married local women and stayed. This would make them relatively recent, not ancient. But genetic dating of the admixture event, the moment when Middle Eastern and South Asian DNA first blended in this population, pushes the founding far deeper into the past. Centuries before Islam existed. Centuries before any medieval trade network could explain them. A fourth theory tried to sidestep biology entirely. Their Jewishness is purely cultural, this argument said. Religion is practice, not blood. There is no biological trace to find. Genome-wide autosomal analysis proved this wrong in a single dataset. The Bene Israel carry a measurable, statistically significant Middle Eastern ancestry component that is completely absent in their non-Jewish neighbors living in the same towns, speaking the same language, eating the same food. That component is not cultural, it is chromosomal. So if none of those explanations work, what does the actual science say? The landmark study came in 2010. A team led by the geneticist Doron Behar, working with collaborators across multiple countries, published a paper in the journal Nature. The study was enormous in scope. Behar's group collected DNA from roughly 200 Jewish individuals representing more than 14 distinct diaspora communities worldwide, from Eastern Europe to North Africa to the Middle East. Each sample was genotyped at hundreds of thousands of SNP markers, single points across the genome where human DNA is known to vary from person to person. The method, in plain terms, was comparison. Take a Jewish person from Mumbai and a Jewish person from Krakow and a Jewish person from Casablanca. Scan their genomes at the same hundreds of thousands of variable positions, then do the same for dozens of non-Jewish reference populations, European, Middle Eastern, African, South Asian, and ask a single question. Do Jews from wildly different countries share a common genetic ancestor? Or did each community arise independently, through local conversion, with no shared biological thread? Crucially, Behar's study included both the Cochin Jews of Kerala and the Bene Israel of Maharashtra. These were among the most anticipated samples in the entire project. Indian Jews had been a marginal interest for decades. Most genetics funding and attention went to Ashkenazi populations in Europe and North America, where samples were abundant and universities were nearby. Collecting DNA in India was logistically harder, and the Indian Jewish population was already shrinking fast, thousands emigrating to Israel every decade. The Bene Israel were a kind of test case for the whole hypothesis. If they showed no Middle Eastern ancestry at all, it would have shattered the idea that Jewish communities around the world share any universal genetic connection. The entire framework would have cracked. Imagine sitting in a lab, opening a file that either confirms or destroys a decade of work. That was the weight on this dataset. When the results finally came back, the signal was not ambiguous. It was not a maybe. The Middle Eastern genetic component in the Bene Israel was clear enough to see on a simple scatter plot, a visible cluster sitting between South Asia and the Levant, belonging fully to neither. But what that cluster meant, and where it pointed, required peeling apart the genome layer by layer. The father's line said one thing, the mother's line said something else, and the combination of both would turn out to be the strangest part of all. The first genomes to come back were the easiest ones. The Baghdadi Jews of Kolkata and Mumbai arrived in India during the late 18th and 19th centuries, and their history is well documented. They came from Iraq and Syria, spoke Arabic, built grand synagogues, and made their livings in trade. Nobody seriously questioned their Middle Eastern origins. They were the control group, and the DNA confirmed it instantly. Baghdadi Jewish genomes clustered almost perfectly with Iraqi and Syrian reference populations on principal component analysis plots, landing roughly 90 to 95% within the Levantine and Mesopotamian genetic neighborhood. The South Asian component in their DNA was negligible, less than 5 to 10%, a whisper after 2 centuries on Indian soil. That number alone is remarkable. 200 years is 8 generations, more than enough time for a population to absorb significant local ancestry through intermarriage. Most immigrant communities do. The Baghdadis did not. They practiced endogamy, marriage exclusively within the community, with a discipline that borders on biological isolation. Generation after generation, surrounded by tens of millions of people, they kept their genetic boundaries almost perfectly sealed. What this proved was methodological, not dramatic. The genotyping worked. If the technology could cleanly separate a known Middle Eastern immigrant group from the Indian populations living beside them, then the method was trustworthy. Whatever signal appeared in the older, more mysterious Jewish communities of India would not be an artifact. It would be real. Walk into the Magen David Synagogue in Kolkata today, and you will find this story's strangest footnote. The building was constructed in 1884, red brick and soaring columns, built to hold hundreds. Now it holds almost no one. The Jewish community of Kolkata has dwindled to single digits. A caretaker who is not himself Jewish unlocks the doors each morning and sweeps the marble floors. The DNA of the Baghdadi Jews points unambiguously to Mesopotamia, but the Baghdadi Jews themselves have almost entirely left India. Their genome is Middle Eastern, their synagogue is a museum, so the recent arrivals checked out exactly as expected. But what happens when you test a community that claims to have been in India for over 1,000 years before the Baghdadis even showed up? The Cochin Jews of Kerala are that community. Their traditions place their arrival either during the reign of King Solomon, when spice traders sailed between the Levant and the Malabar coast, or after the destruction of the Second Temple by Rome in 70 CE. Either claim, if true, would make them among the oldest Jewish diaspora communities anywhere. And their father line pointed to the ancient Middle East. But DNA has two parents. The question nobody wanted to ask out loud was simple. What about their mothers? Mitochondrial DNA tells the maternal story. It passes from mother to daughter to granddaughter in an unbroken chain, never mixing with the father's contribution, carrying its own separate record of ancestry. When researchers sequenced the mitochondrial DNA of Bene Israel women, the answer was immediate and unambiguous. Their maternal haplogroups were South Asian, not partially, not ambiguously, overwhelmingly. The markers that appeared, M, R, U2, are the deep maternal lineages of the Indian subcontinent, shared with Maharashtrian Hindu women, shared with populations across western and central India. The haplogroups that define Jewish women in other diaspora communities, K, N1b, H, HV, lineages rooted in the ancient Near East, were almost entirely absent. 80 to 90% of Bene Israel maternal lineages traced to India. Fewer than 10% showed any possible Middle Eastern or ambiguous origin. The implication was stark. The men who founded this community came from the Levant. The women they married were already here. This was not a whole community migrating together, families intact, from one land to another. A small group of Jewish men arrived on the Konkan coast, married local Indian women, raised their children in the Jewish faith, and then the community sealed itself shut. Endogamy took over. No more outsiders in, almost no one out. For centuries, there is a deep irony buried in this finding. Traditional Jewish law, halakha, defines Jewishness through the mother. If your mother is Jewish, you are Jewish. That is the rabbinic standard. Yet genetically, the Bene Israel's Jewish ancestry came almost entirely through their fathers. The mothers were Indian. By the strict matrilineal rule, the founding generation would not have been considered Jewish at all. Some scholars believe this is not a contradiction, but a clue about timing. The matrilineal standard may not have become normative until the Mishnaic period, around 200 CE. If the Bene Israel's founding predates that shift, they would have operated under an older patrilineal model. Their DNA may be recording a version of Judaism older than the rules most modern Jews follow. Picture a home in Thane, a city pressing up against Mumbai's northeastern edge. A Bene Israel woman lights Sabbath candles at a wooden table. Her mitochondrial DNA traces to the same deep Indian maternal lineage as her Hindu neighbor 2 doors down. But her sons carry a Y-chromosome stamp from the ancient Levant. 2 continents live inside one family. You cannot see it in their faces. You can only see it under a sequencer, so the Bene Israel have Jewish fathers and Indian mothers. But the Y-chromosome and mitochondrial DNA together account for less than 2% of anyone's total genome. They are 2 narrow lines of inheritance in a vast genetic landscape. The real question remained. How much of the Bene Israel's full genome, all 22 non-sex chromosomes, the complete autosomal picture, is actually Middle Eastern? The answer came from genome-wide autosomal analysis, the kind of scan that reads not just a single parental line, but the entire inherited mosaic. Every person's autosomal DNA is a shuffled blend of all their ancestors, maternal and paternal, going back thousands of years. When researchers ran this analysis on Bene Israel samples, The Middle Eastern contribution did not vanish into noise. It held. Estimates place the Levantine or Middle Eastern ancestry component at roughly 20 to 35% of the Bene Israel genome. The remaining 65 to 80% was South Asian. Those numbers deserve a pause. 20 to 35% is not a trace. It is not a statistical ghost. After 1,500 to 2,000 years of living in India, marrying within the community, absorbing no further Middle Eastern input, nearly a quarter to a third of their genome still pointed to the Levant. That survival rate meant the community's marriage boundaries had held with extraordinary discipline across at minimum 60 generations. When you plot this data visually, the picture becomes even more striking. On principal component analysis plots, which map genetic similarity across 2 dimensions, every tested population forms a cluster. South Asians form one cloud. Middle Eastern Jews form another. The Bene Israel sit between them, pulled visibly toward both, belonging fully to neither. No other tested population on Earth occupies that exact intermediate space. They are, genetically, a bridge between 2 continents. Imagine a researcher's laptop in a university office, a scatter plot filling the screen. Thousands of gray dots for Indian reference populations grouped on the left. Hundreds of blue dots for Jewish populations grouped on the right. And in the middle, a small, tight knot of Bene Israel data points, suspended alone. A gradient also emerged when comparing India's 3 Jewish communities side by side. The Baghdadis, who arrived 2 centuries ago, sat at roughly 90% Middle Eastern. The Cochin Jews, who claim an ancient presence, showed a somewhat lower Middle Eastern share. The Bene Israel fell between them. The gradient tracked perfectly with each community's claimed age of arrival. The longer a community had lived in India, the more South Asian DNA it carried. Time itself was visible in the data, but one number was still missing. The DNA said the Bene Israel were part Middle Eastern, part Indian. No question. It did not yet say how many people started the whole thing. Founder effect analysis attempts to answer exactly that. By measuring the diversity within the Bene Israel's Middle Eastern genetic component, researchers can estimate how many original founders contributed it. A large founding group leaves behind a wide spread of genetic variants. A tiny one leaves a narrow bottleneck, a few branches on what should be a sprawling tree. The Bene Israel's Middle Eastern component showed the bottleneck signature. The estimated number of founding males was staggeringly small, as few as 4, perhaps as many as 8. Not 400, not 40. 4 to 8 men of Middle Eastern origin who reached the Konkan coast and married local Indian women roughly 1,600 to 2,000 years ago. That time window places them in the late Second Temple period, possibly in the immediate aftermath of Rome's destruction of Jerusalem in 70 CE. Scale that forward. 4 to 8 men become 20,000 descendants over 60 to 80 generations, roughly 2 millennia at 25 years per generation. A population the size of a small town, grown from a group you could seat at a single dinner table. And here is the detail that turns an already extraordinary finding into something eerie. The Bene Israel's own oral tradition, passed from parent to child for centuries, says that exactly 7 couples survived a shipwreck off the Konkan coast near Navgan. 7 couples. The genetic estimate says 4 to 8 founding males. An origin myth dismissed as legend by every serious historian for 200 years turns out to be approximately correct. Stand on the coast near Navgan today, south of Mumbai, where the tradition places the wreck. The shore is rocky and monsoon-battered, wave-cut platforms slick with spray. A local guide will point at the water and tell you the story as his grandfather told it to him. Somewhere beneath that surface, or woven into the local gene pool, the evidence of those original founders persists. A tiny group of men, a shipwreck story that might be true, and 2,000 years of isolation. But here is what nobody expected. This exact same pattern, the same genetic formula, was already hiding inside Jewish communities on the other side of the world. 6 different categories of evidence. Paternal lines, Middle Eastern. Maternal lines, South Asian. Whole genomes, 20 to 35% Levantine. Founder effect models, 4 to 8 men. Genetic clocks, 1,600 to 2,000 years. An oral tradition older than most nations, 7 couples from a shipwreck. They all land on the same spot. This is no longer a mystery about India. It is a pattern. The hidden genetic origin of the Indian Jews is not hidden anymore. The Bene Israel are genuinely descended from a small group of ancient Jewish men, almost certainly from the Levant, who reached India's Konkan coast roughly 1,600 to 2,000 years ago. They married local Indian women, they founded a community, they closed their marriage borders, and they maintained Jewish practice in near total isolation for over a millennium, possibly closer to two. They are not converts. They are not medieval arrivals. They are not a cultural borrowing from a passing merchant fleet. Their DNA carries a measurable, verifiable stamp of ancient Middle Eastern paternal ancestry that no amount of cultural study, no archival research, no linguistic analysis could have confirmed or denied on its own. It took a genome to settle it. The State of Israel formally recognized the Bene Israel as Jews in 1964, after years of rabbinical debate and bureaucratic hesitation that had real consequences for real families trying to immigrate. Genetics, decades later, reached the same conclusion by an entirely separate road. No scripture, no testimony, no oral argument, just base pairs. The DNA does not suggest it, it confirms it, and that, on its own, would have been enough to close the case. But the data did something else, something the researchers were not looking for. It exposed a much larger secret. The Bene Israel's genetic structure is specific. Middle Eastern fathers, local mothers, a tiny founding male population. Deep endogamy, sealing the mixture in place for centuries. That structure is not unique to India. It is the template for almost every major Jewish diaspora community on the planet. Start in Europe. Genome-wide studies of Ashkenazi Jews, the largest and most studied Jewish population on Earth, have shown a strikingly similar architecture. A 2013 study led by Marta Costa and published in Nature Communications analyzed Ashkenazi mitochondrial DNA in detail and found that a large proportion of Ashkenazi maternal lineages traced to Europe, likely Southern Europe, possibly Italian women who married Jewish men during the Roman period. Meanwhile, Ashkenazi paternal lineages are predominantly Middle Eastern. The formula is the same. Jewish men from the Levant, local women from the host population, endogamy closing the door behind them, moved to the Iberian Peninsula. Sephardi Jews show a parallel signature. Their maternal lineages carry Iberian and North African contributions. Their paternal lines point back to the Middle East. Same formula, different continent. Now crossed to East Africa. Ethiopian Jews, the Beta Israel, carry significant East African maternal ancestry. Their Y-chromosomes show markers linked to Middle Eastern populations, though the strength of this signal is debated, and the Ethiopian case is genetically more complex than the others. Even so, the underlying architecture rhymes. Male founders from the Levant, local mothers from the surrounding population. A community sealed by endogamy, the same script, running on 4 continents. Think about what this means. The Bene Israel were not an anomaly. They were not a strange outlier, an exotic footnote in Jewish history that happened to produce a quirky genetic result. They were one instance of a global founding mechanism. Across 2,000 years and thousands of miles, small groups of Jewish men dispersed outward from the ancient Middle East. They settled in distant lands. They married local women. They converted their families and raised their children Jewish. And then they sealed the community shut, generation after generation, maintaining genetic and cultural boundaries that most populations dissolve within a few centuries. The Indian Jews did not just reveal their own origin story. They accidentally exposed the genetic blueprint that built almost every Jewish community on Earth. The same small group of men, the same local wives, the same closed door behind them, repeated on every continent. Picture a map of the ancient world with arrows radiating outward from the Levant. One arrow points to Rome, another to Iberia, another south to Ethiopia, another east across the Arabian Sea to the Konkan coast. At the tip of every arrow, the same genetic formula appears. Jewish Y-chromosome plus local mitochondrial DNA plus endogamy equals a new diaspora community. The Bene Israel did not know they were part of this pattern. Neither did the Ashkenazim, neither did the Sephardim. It took a genome-wide comparison of all of them, side by side, to see that every Jewish diaspora had been born the same way. And the evidence does not rest on DNA alone. Independent confirmation arrives from entirely separate categories. Linguistically, the Bene Israel historically spoke a distinctive form of Marathi that contained Hebrew loanwords absent from standard Marathi. These were not Arabic borrowings, which would suggest contact with later Muslim-era traders. They were Hebrew terms for religious practices, calendar features, and specific foods. Vocabulary that implies direct transmission from Hebrew-speaking ancestors, not second-hand cultural diffusion through a chain of intermediaries. The language itself carries a timestamp. Words borrowed from Hebrew before Arabic became the lingua franca of the Middle East, point to a founding population that left the Levant before the 7th century. Archaeologically, the trail runs deeper still. In Kerala, ancient copper plates record land grants given to a Jewish leader named Joseph Rabban by a local ruler. These plates have been dated to somewhere between the 4th and 10th centuries CE, depending on the scholar. But even the most conservative estimate places an established, officially recognized Jewish community in India centuries before any medieval migration wave could explain them. Joseph Rabban did not arrive from Baghdad in the 1800s. He was already there, already known to local kings, already holding title to land. There is also a second genetic line of confirmation. Populations that pass through a tight founder bottleneck do not only carry distinctive ancestry proportions, they carry distinctive disease risks. When a tiny group founds an entire community, any rare genetic variants those founders happen to carry become disproportionately common in all their descendants. This is why Ashkenazi Jews have elevated rates of Tay-Sachs disease, and why certain Finnish populations carry rare metabolic disorders at unusually high frequencies. The Bene Israel show a parallel pattern, elevated rates of specific autosomal recessive conditions, found more frequently in Middle Eastern and Jewish populations than in surrounding Indian groups. The bottleneck left its fingerprint not only in ancestry percentages, but in the specific medical vulnerabilities the community carries. Language, royal land grants, and disease genetics. 3 categories of evidence that have nothing to do with SNP arrays or principal component analysis, and all 3 converge on the same conclusion the genome-wide data already reached. Ancient Middle Eastern founders, not modern converts, not medieval merchants. Not a cultural accident. The specific mechanics matter here, so let us go one layer deeper into the chromosomes. The Y-chromosome marker that links Bene Israel men to the Middle East belongs to haplogroup J, and specifically to its 2 major subdivisions, J1 and J2. These are not obscure categories. J2 is one of the most common paternal lineages in Levantine, Anatolian, and Mesopotamian populations. It appears at high frequency in Jewish men across diaspora communities worldwide, from Warsaw to Casablanca to Sao Paulo. J1 is closely associated with Semitic-speaking populations, both Arab and Jewish, anchored in the Arabian Peninsula and the Fertile Crescent. In the Bene Israel, these haplogroups appear at rates far exceeding anything found in surrounding non-Jewish Indian populations. Among Marathi-speaking Hindu men in the same region, the dominant Y-chromosome lineages are completely different. Haplogroups H, L, and R2, signatures of deep South Asian ancestry. The contrast is not subtle. It is 2 different paternal worlds living side by side. The autosomal data adds another dimension of precision. Using admixture software, a standard tool in population genetics, researchers decompose each person's genome into components representing hypothetical ancestral populations. The number of assumed ancestors, called K, is increased step by step to see how the data splits. At every K value tested, the Bene Israel genome consistently breaks into 2 primary components. One aligns with Levantine and Middle Eastern reference populations. The other aligns with South Asian groups. That alone is significant, but at higher K values, something more specific emerges. The Middle Eastern component in the Bene Israel does not cluster with non-Jewish Middle Eastern populations like Palestinian Arabs or Bedouins. It clusters specifically with other Jewish groups, Ashkenazi, Sephardi, Mizrahi. This is not generic Middle Eastern ancestry. It is Jewish-specific genetic drift, a shared history of isolation and endogamy that left a distinctive stamp on the DNA of every Jewish community it touched. The Bene Israel carry that same stamp. One final signature confirms the endogamy from the inside. The Bene Israel show long runs of homozygosity, stretches of the genome where both copies of a chromosome are identical, inherited from a common ancestor on both the mother's and father's side. Long runs of homozygosity are a hallmark of populations that have married within themselves for centuries. The Bene Israel's profile sits in an intermediate range, longer than the runs found in outbred Indian populations, but shorter than the extreme homozygosity seen in Ashkenazi Jews, whose endogamy was tighter and lasted longer in a smaller European population. The position fits perfectly, endogamous, but not maximally so. Isolated, but within a subcontinent large enough to buffer the effect. 2,000 years of marrying your neighbors, and the genome recorded every generation of it. There are things this evidence does not tell us. Genetics cannot explain why these men traveled to India. Trade, flight from Roman persecution, military service, a shipwreck, or something no record preserved. The motive is invisible in the DNA. The oral tradition of 7 couples surviving a wreck is consistent with the genetic estimate of 4 to 8 founding males, but consistency is not proof. Founder effect models carry wide confidence intervals, and the number 7 may be coincidence rather than memory. Sample sizes for Indian Jewish genetics remain small compared to the vast data sets available for Ashkenazi or Sephardi populations. And the Cochin Jewish community in India has dwindled to fewer than 30 people, making further sampling nearly impossible. The genetic clock that dates the admixture event gives a range, roughly